Conclusion: Late-chain amyloidosis. Amyloidosis disease is caused by the deposition of normally soluble in whey proteins in the body. Amyloid proteins accumulate in the tissues and organs. In consequence, they change their shape and disorder their work. The video.
Amyloidosis is the name for a group of rare diseases in which abnormal proteins misfold, leading them to aggregate and deposit in tissues and organs where they cause damage. Proteins are molecules that are essential for the structure and functioning of all living organisms. Our body produces many thousands of different proteins. Each has a specific role and is made according to the genetic instructions carried within our DNA. Once made, the protein folds into a particular shape, allowing it to work properly and interact with other molecules. Sometimes proteins misfold and don't function properly, but normally our body is very good at getting rid of them before they cause any problems. Problems can occur if, for example, the removal mechanism isn't working properly, or if more misfolded proteins are made than our body can handle. Amyloidosis occurs when misfolded proteins aren't removed in the normal way. Instead, they accumulate and stick together to form long, rigid fibrils that deposit in various tissues and organs. These deposits are called amyloid, and over time, start to cause damage. More than 30 different proteins are known to misfold and form amyloid, meaning that over 30 different types of amyloidosis exist. Some proteins misfold because of a faulty gene we inherit from one of our parents, whereas others misfold because of factors we acquire during the course of our lives, or as a result of aging. Regardless of the type, once formed, amyloid is insoluble and difficult to break down. As it builds up, it stops the affected organs from working properly. Symptoms vary depending on which organs are involved. The most common include the heart, kidneys, liver, spleen, nervous system, and digestive system, but this varies according to the type of amyloidosis and also differs between patients who have the same type. There is currently no cure for amyloidosis, but treatment options are available for the most common types, and novel treatments are being investigated, including ways to stop misfolded proteins being made and ways to stabilize proteins to prevent them from misfolding. The aim is to stop disease progression, restore organ function, and improve quality of life for patients.
The classification based on the cause, primary, secondary, based on extent of amyloid deposition, systemic or localized, and 3 based on type of amyloid. Types of amyloidosis: the LA amyloidosis, primary, caused by deposition of light-chain proteins produced by bone marrow. AA amyloidosis, secondary, it's a result of chronic inflammatory disease caused by accumulation of SAA protein. Amyloid type A protein is most commonly affects the spleen, kidneys, liver, adrenal gland, and lymph nodes. ATTR amyloidosis, hereditary form, caused by accumulation of abnormal transitin TTR, which is made in the liver. A amyloidosis, senile form, common in older men. DRA amyloidosis, dialysis-related amyloidosis, caused by long-term and beta microglobulin can't be entirely removed from the body. LA amyloidosis, result of excess production of light chains immunoglobulins by transformation of plasma cells. Those light chains come together and form normal amyloid fibers, which can't be destroyed in proteolysis, so they go through the bloodstream and accumulate in tissues and organs. The most affected organs are kidneys, liver, heart, and intestines, peripheral nerves.
Amyloidosis is the term used to describe a group of diseases in which deposits of an abnormal protein called amyloid form in tissues and organs throughout the body. Proteins are important molecules involved in almost every structure and function within our cells. They are made up of chains of smaller units called amino acids, which fold themselves into three-dimensional structures. Folding allows the protein to work properly and interact with other molecules. In amyloidosis, proteins are made in the normal way, but when they are released from the cell, they open up again. These abnormally folded proteins collect together in long chains or fibrils. The deposits they form are called amyloid. Amyloid fibrils are difficult to break down, and as they accumulate, they start to cause damage. Eventually, the buildup of amyloid fibrils stops the affected tissue from working properly. AL amyloidosis is caused by a bone marrow disorder. The bone marrow produces blood cells, including plasma cells, a type of white blood cell that forms part of the immune system. Plasma cells make antibodies to help fight infection. Each antibody is made up of 4 protein chains, 2 heavy chains and 2 light chains, which attach to each other to form the complete antibody. In AL amyloidosis, the abnormal protein that builds up and causes problems is the antibody light chain, hence the name AL amyloidosis. Instead of attaching to heavy chains, the abnormal light chains open up and bind to other abnormal light chains to form amyloid fibrils. These gradually build up over time. Amyloid light chain fibrils can occur almost anywhere in the body, but the organs most commonly affected include the kidneys, the heart, liver, spleen, nerves, and the digestive system. Treatment for AL amyloidosis involves chemotherapy, steroids, high-dose therapy, and stem cell transplant, and other drugs. Treatment is aimed at reducing the plasma cells to stop further amyloid light chains being made that would otherwise build up in tissues and organs. It also gives the body a chance to break down existing deposits naturally. However, on its own, this process can take many months or years. New treatments which break down the amyloid deposits more quickly are currently being tested in AL amyloidosis. The hope is that they can reduce symptoms and restore function quickly, and in doing so, improve quality of life for patients.
Diagnosis. Biopsy of the tongue was performed and the findings confirmed a diagnosis of late amyloidosis, which appears under polarized microscopy as an apple-green birefringence after Congo red strain. Treatment in amyloidosis has not occurred. Supportive therapy, diet, low-salt diet, pain medication, limited further production of amyloid proteins, if it is possible. Chemotherapy, cell transplantation, AA, we can treat the underlying disease, TTR, liver transplantation. Conclusion: amyloidosis is a well-recognized cause of macroglosia. Early diagnosis and treatment are critical to prevent irreversible organ damage. This patient died shortly after the diagnosis was made as a result of complications from aspiration and pneumonitis.